Welcome to video 3 of IB Biology Topic 3 Genetics. In this video we are looking at section 3.3 Meiosis. Okay, so your essential idea behind this topic is that our alleles, which we looked at in section 1, where we were looking at genes, and we said that on our chromosomes we had genes that coded for various traits, and on a pair of chromosomes obviously we've got two genes, but they can be different, coding for the same trait, but they can be in a slightly different form, and we said that was due to the change in a base or a manipulation of that base sequence, which basically meant that different trait, so if you had blonde hair and brown hair, one of them was actually a mutation. Okay, and we said these different genes, so coding for these same traits but different characteristics as such, we called them alleles. So the essential idea of this unit is that these alleles segregate during meiosis, allowing new combinations to be formed in the fusion of gametes. So you would remember cell division of mitosis from your earlier years of study, and you would have already covered meiosis as well, but now it's time to do it again and in a little more detail. So in your IB, you have the area of your nature of science. And for this unit, this section, we'll be looking at making careful observations. Okay, so what you see, or what is seen. So meiosis was discovered by microscope examination of dividing germline cells. Okay, so this links us back to section 1.8 at the start of our course. Now, so meiosis was discovered by microscopic observations way back in the 1800s. This was made possible with the improvement in microscopes and the discovery of dyes that could stain these thread-like structures, which we now know as chromosomes, in the nucleus. A key observation that in the horse threadworm, there were 2 chromosomes in the nucleus of an egg and sperm cells, whereas the fertilized egg contained 4. So they're looking at these cells from the threadworm of a horse, and they noticed that in the gametes there was only half the genetic material. However, in the fertilized egg we had the full set of genetic material. So they were those early observations that built the knowledge that we have today. This indicated that the chromosome number is doubled in fertilization. Observations then led to the hypothesis that there must be a special nuclear division in every generation that halves the chromosome number. So they knew that cells divided, however, they were unaware of the fact that there must be this separate division that occurred to make the gametes. The sequence of meiosis was eventually worked out by observing cells taken from the ovary of rabbits. So now we'll move on to looking at some of our areas of understanding. Now, first of all, we are looking at one diploid nucleus dividing by meiosis to produce these 4 haploid nuclei. So remembering our terms, diploid being the full set of genetic information and haploid being that half set, so that those haploid cells fuse during fertilization to produce one diploid cell. And here it is. Look at it in its full splendor. One diploid cell at the start, going through meiosis to eventually form 4 haploid nuclei. And by the end of this unit, you are going to be able to draw this diagram yourself and understand each stage. And here we've got the process again in another infographic. People like these in different ways, so giving you a variety of images will help you to choose what kind of image suits you. what works best for your learning. Okay, the halving of the chromosome number allows a sexual life cycle with the fusion of gametes. So basically what we're saying is, if the diploid cells fertilize, that would give you a completely different species. Yeah. So we need that halving of the chromosome number to occur to allow the fusion of gametes to allow a new reproductive being to be created. So what happens? Well, DNA is replicated before meiosis so that all the chromosomes will consist of 2 sister chromatids. So we've got a lovely infographic here. This is one I'd really be drawing down, just so you're really remembering this terminology. One chromosome consists of 2 sister chromatids, and we've got the centromere joining them in the center there. So DNA is replicated during the synthesis stage of the cell cycle during that interphase. So we're going way back here to unit 1.6, 1.8, where we looked at that mitosis cell cycle. After replication, the chromosomes appear as 2 sister chromatids held together by a centromere. Perfect, that's that image there. Okay, we talked about this in the previous video, this whole concept of one chromosome. Okay, so our chromosome is double-stranded 2 DNA molecules which have those 2 identical chromatids. So we're at that stage at the start of mitosis, okay? When they're about to divide during mitosis or the anaphase of meiosis, so that's what we're saying, we've got our 2 chromatids. Now it's single-stranded, 1 molecule of DNA after cell division has occurred. So just remembering when we have our double-stranded and our single-stranded chromosomes. So when we've got double-stranded, that's when we've got that chromatids, our 2 sister chromatids ready for cell division. These graphs are just showing you what's happening in the cell. So the top graph, we can see on our vertical axis, we've got cell volume, and on our horizontal axis, we're looking at time. And we can see as time increases there, that cell volume is increasing until something happens and that cell volume decreases. So that cell volume would decrease at cell division, and then the whole cycle occurs again. If you look at the bottom graph, you can see we've got the different stages of the cell cycle on the horizontal axis, and on the vertical axis, we've got the amount of DNA. And you can see how during the S phase there, our amount of DNA increases, stays constant during G1, G2, drops during M, okay, and then constant for a period during the first cell cycle, and then we see the same pattern repeating and repeating, okay? Again, with those kind of graphs, it's important that you can read the story of the graph as such to say what the graph's showing. Now, the early stages of meiosis involve pairing of homologous chromosomes and crossing over, followed by condensation. So I'm sure you understand all of these terms and what this actually means. This should just be reminding you of studies from earlier in your schooling. So we're in meiosis 1 at the moment, and we're in the prophase. Okay, now during prophase 1, now remembering meiosis has 2 phases as such. So we have meiosis 1, meiosis 2, which means we have the prophase 1, prophase 2. Okay, so during prophase 1, the chromosomes condense and become visible. You can only see chromosomes, remember, once they're condensed. Before then, they're this spaghetti mess. Okay, now homologous chromosomes, known as homologules, they pair up. Crossing over occurs. Now this process of crossing over we will look at a bit further in the following slides, but this is where chromosomes actually swap some of their genetic material. It's quite fascinating. Also during prophase 1, our spindle fibres are being produced at the microtubule forms, okay? So, homologous chromosomes, first of all right, these are where we've got 2 chromosomes of the same type. All right, and when you've got two of them together, each is called a homologule. So we've got our chromatids, which is each strand, our chromosome, which is the whole unit joint with the centromere, and then homologous chromosomes are two of the same chromosomes. Each chromosome is called a homologule. So just getting some of that terminology. Okay, so this picture here, we're looking at two homologous chromosomes that are nicely aligned. Now, it's showing us that chromatid crossover. So you see how they kind of swap a part of the chromosome, and then you've got some genetic variation occurring here because you've got some of the blue on the red and some of the red on the blue as such. Again, another image of chromosomes there. Now, we call this bivalent formation. So two, bi meaning two. Okay, so two chromosomes align in bivalent. So the early stages of meiosis involve pairing of homologous chromosomes to create bivalents, so pairs. This is followed by condensation, so where they become condensed so they're visible. As DNA replication has already occurred, the chromosomes already consist of their double chromatids. Okay, so you've got your chromatids. A homologous pair is bivalent, so homologous pair means two, bivalent, two of the same, and consists of 4 strands of DNA, 2 making up each chromosome. That's all good. I'm sure you're all good with that. Oh dear, my graphics are not good. Okay, while paired crossing over occurs where there is an exchange of genetic material between non-sister chromatids, okay, of the homologous pair. That's what we mean by crossing over. Pieces of chromatid break off and rejoin the other chromatid. We've looked at that. This happens in at least one place on each homologous pair. Can occur randomly in other places, especially for our larger chromosomes. And a mutual exchange of genes occurs between the chromosomes, resulting in these new combinations. Okay, so this this great variation occurring in the alleles. So again, this is just another image to show that crossing over occurring and what the product is from there, showing the probability of different combinations of genes. Okay, so that's prophase 1. Now let's move on to the metaphase 1. During the first metaphase, our homologous chromosomes have lined up along the equator. The nuclear membrane is now disintegrated, and the fibers then contract and pull the homologous pairs apart. So we've got the fibers have joined to the chromosomes. Now they're contracting and pulling our homologous pair apart. Now, remembering that our chromosomes have lined up along that equator, and we call this random orientation of bivalence. So we know our bivalents are our homologous pairs. Random orientation means exactly what it says, that random orientation. So the spindle fibers, they attach to the centromere of each of the chromosomes. Each chromosome is attached to one pole only, not both. So the two homologous chromosomes are in bivalence, so we've got our pair. Each are attached to different poles. The pole to which they attach depends on the way the pair is facing or orientated. So the one closer to the pole will be what that pole connects its fibre to. There's no going around catching the one on the other side, they just connect to the closest. The orientation of the bivalents is completely random, so it's random which pole they're going to be pulled to. And the orientation of one bivalent does not affect any of the other bivalents, so it is completely random how they all align. Results in the random assortment of maternal and paternal chromosomes, because it is completely random each cell. Okay, so that was the metaphase 1. Now we're moving on to the anaphase 1. And during the anaphase, these spindle fibers from the pole attached to our chromosomes, and these fibers are then contracted and pull apart. So the real pulling apart occurs now during the anaphase. Okay, this is where they're really separated out. So you have some of the connection, and that is occurring towards the end of the metaphase and really happening during the anaphase, and we pull those homologous pairs apart. And then during the telophase, so telophase 1, remember, we're still in meiosis 1, those spindle fibers disintegrate, spindles disintegrate, the chromosomes uncurl, and new nuclear membranes form. Many plants do not actually have a telophase 1, okay? They just go straight into meiosis 2. Okay, so here we're looking at this image of meiosis 1, just showing you in a pictorial way what is occurring, and remembering that one of your skills is to be able to draw the full process of meiosis. Another image here, I actually much prefer this image myself, but again it depends on you and your preferred way. It's really important that during your drawing of meiosis that you do show the crossing over, and you do keep that quite visual throughout your images. Okay, so after meiosis 1, cytokinesis occurs. So the cell splits in half, and at this point the daughter cells are haploid because they contain only one chromosome from each pair. However, each chromatid still has their sister attached, so no S phase is now required. So the point of meiosis 1 is separates the homologous pairs, resulting in 2 haploid cells. This is the first division, and it's sometimes called a reduction division. Alrighty, well, let's go on to meiosis 2 and see what's occurring there. So, meiosis 2, we've got our prophase 2 and our metaphase 2. So in the prophase 2, the DNA is condensing again to become visible chromosomes. New meiotic fibers are produced. Metaphase 2, our nuclear membrane breaks down, chromosomes line up along the equator of each cell, and our spindle fibers from opposite poles attach to the chromatids at the centromeres. Our anaphase 2. Again, during the anaphase, these centromeres of each chromosome split, releasing each sister chromatid. So in the last one, we had our homologous pairs being divided. Now we're getting the chromatids divided. The spindle fibers pull individual chromatids to the opposite ends of the cell. Due to the random orientation, the chromatids could be pulled towards either end of the newly forming daughter cells. Now in animal cells, the cell membranes then pinch in the middle. In plant cells, new cell plates form to separate the 4 cells. Okay, so here we are now in our telophase 2. Telophase 2. Chromosomes unwind, nuclear envelopes form around each of the 4 haploid cells, preparing them for cytokinesis. And there we have meiosis 2. Okay, so remembering you need to be able to draw the full process of meiosis. This is just showing you meiosis 2, which shows you from the telophase and cytokinesis through meiosis 2. So the role of meiosis 2, or the purpose, is to separate the sister chromatids, creating new single-stranded chromosomes. And this then results in our 4 haploid cells, which we know as gametes, sperm or egg cells. So why does crossing over and random orientation occur? It is because it promotes our genetic variation. Not only have we got different parts of the chromosomes then being on different chromatids, we also have this random arrangement. So it's literally impossible to have exact genetic representation in each cell, in any cells. That then increases this whole concept of variation that we looked at in our evolution unit. Fusion of different gametes from different parents then promotes this even further, because we've got this process occurring in both parents. Okay, so the variation is extreme. Okay, so your applications and skills, where do we take this information now? First of all, non-disjunction can cause Down syndrome and other chromosomal abnormalities. So what is this whole concept then of non-disjunction? So normally at meiosis, the cell divides into 4 haploid cells, where each cell contains 1 homologule. Okay, now non-disjunction is when separation of chromosomes fails to occur. So if you look at the image, you can see that one of our gametes actually has no chromosomes in it, and one has 2 chromosomes in it. So when you're looking at the blue chromosome, okay, we've got 2, so an extra chromosome in the gamete there. And here we've got an image, so you can see during the metaphase there on the left hand side. When you look at during the anaphase in normal disjunction, you can see how the chromatids are pulled apart, but in non-disjunction you can see they're not actually pulled apart and both have gone to one side. Now this can occur at both stages of the of both anaphases, and that causes different outcomes, so we will look at that further in a moment. That's what this image is showing us here. Okay, so this can either happen in the first division, anaphase 1, when homologous pairs are pulled to the same pole. So if you have a look in meiosis 1 in the top diagram, you can see that 2 homologous pairs have been pulled to the top pole. If it happens during the second division, anaphase 2, when the chromatids fail to separate and end up at the same pole. Okay, so here we've got normal disjunction or non-disjunction, and here you can see you've got the 2 over there. So when you look at the outcomes, you can see here we've got 2 single chromosomes, but here we've got 3 chromosomes. Okay, so you've got your N+1 or you've got your N-1s. Okay, here we've got 2 normal cells, so 2 Ns, so these have got 1 less, and here you've got 1 with 1 less and 1 with 1 extra. Looking at that again, okay, so if meiosis occurs at that first junction, you end up with 2 gametes with 24 chromosomes, okay, and you end up with 2 gametes with 22. Okay, so gametes missing a chromosome, they generally do not survive. So you've got 2 with 24, and then you've got 2 with 22. So 2 with an extra and 2 with 1 less. So if you've got 1 less, generally those chromosomes will not survive. If you've got 1 extra, they will, but there'll be a chromosomal they can survive. There'll be a chromosomal abnormality because you've got an extra chromosome there. If non-disjunction occurs in the second division, okay, then the result will be 2 normal haploid cells. So there's our 2 normal. 1 cell that has 1 extra, so you've got 1 extra in that one, and then you've got 1 cell with 1 chromosome missing. Okay, so you would have heard of the condition Down syndrome. Okay, this is trisomy, so 3 somatic. That's what trisomy means, 21. So if you look at chromosome 21, there is the extra chromosome, and that is because of this. Let's go back and have another look back here. That is because of this non-disjunction. So it would be from one of those cells, or one of those cells, you've got 3 chromosomes. another application. Studies showing age of parents influences chances of non-disjunction. So studies have shown that a lot of genetic conditions around disjunction do become more common as the age of the maternal gametes increases. Okay, so we will look at this further in class, but this is a correlation with Down syndrome and maternal age. Your next application, description of methods used to obtain cells for karyotype analysis, e.g. chorionic villus sampling and amniocentesis, and the associated risks. So we'll look at this much further in class, but what we're actually looking at here is genetic testing of embryos, and you can see in the picture that this is where embryo fluid, amniotic fluid, or chronic villus tissue, which is taken from around the placenta, is removed and tested to see for genetic conditions. And this is another image here showing that. Okay, and we will look at this further in class, but the whole idea is tissues removed either from the amniotic fluid or from the chorionic villi, and they're then tested, okay, to see the karyotype of the embryo and see if there's any genetic conditions. All right, a skill. So as I've spoken about throughout the video, one of your skills is to be able to draw the stages of meiosis, which results in the formation of our 4 haploid cells. I don't know why they come up back to back. So we've got our meiosis 1 and meiosis 2. I just like these 2 images myself. Okay, and you need to be able to draw each stage of that. We've talked about disjunction and genetic conditions, and this is just looking at one chromosome here. It's chromosome 12 with 132 million base pairs in it, and it's just showing all the genetic conditions associated with that. Not all of these would come due to disjunction, okay, but I'm just showing you one single chromosome and the conditions that could be connected to it. Okay, so we are looking at some pretty intense conditions and a really, really complicated area, and there is so much study and future here for you to potentially move into in your future careers. Now, just a link here to your theory of knowledge, okay? In 1922, the number of chromosomes counted in a human cell was 48. This remained the established number for 30 years, even though a review of photographic evidence from the time clearly showed that there were only 46. Thinking in your TOC headset now. For what reasons do existing beliefs carry that certain inertia? Okay, thank you for watching video 3, and I'm looking forward to seeing you in our 4th video of the genetics unit.